students will bring in with the last concept of the topic cell cycle and cell division. So in this, yesterday we have dealt with the meiosis process, what all happens during the process of meiosis, that is there are two nuclear divisions, meiosis 1 and meiosis 2, and what are the stages. All the stages are similar to each other with slight changes in the chromosome structures or the features, and we have seen that in mitosis 2 daughter cells are formed while in meiosis 4 daughter cells are formed. So already we have dealt what are the significance of mitosis. Now we will go with the significance of meiosis. What are the significance of meiosis? Earlier, I said in the earlier classes that meiosis occurs in the reproductive cells, or the reproductive organs have particular reproductive cells or the germ cells which undergo this process. Those cells are called as meiocytes, which undergo the process of meiosis to form the daughter cells, that is they help in the formation of the gametes. Gametes in the sense those are the reproductive cells which undergo the process of reproduction, where there are male gametes and the female gametes. So this meiosis is necessary for formation of gametes in animals and spores in plants. So this process is necessary for the formation of the gametes. In animals, that is the sperm and the ova, yes, while in the plants they are in the form of spores. Example, you take the pollen grains, which are the reproductive organs. So this process helps in the formation of reproductive cells. Yes, reproductive gametes, or we can say the spores. In gymnosperms, we have studied about the microspores and the megaspores. Microspore being a male gamete and megaspore being the female gametes. So this process is necessary for the formation of the gametes in animals, while in plants it is helping in the formation of the spores. Here, it also helps to maintain, it maintains the number of chromosomes which are haploid in condition. They are maintaining, it helps to maintain the number of chromosomes. By the end of the process of meiosis, 4 daughter cells have been formed. Yes, that is the gametes that have been formed are haploid, that is they have only half the number of chromosomes. So these gametes, when they fuse together, they develop to form a zygote, then into an embryo, while the embryo have 46 number of chromosomes. So that is continued as we are getting the chromosomes from the paternal and the maternal side. So each and every adult organism have diploid number of chromosomes, but the gametes have haploid number of chromosomes. So it is helping to maintain the number of the chromosomes to be haploid, that they are haploid. So this process, if throughout, let it be any form of organisms, all the gametes that are being produced are haploid in number, thus it helps to maintain the number of the chromosomes. Why? We have gone through the process like crossing over. Crossing over exchange of segments leads to variation, which is a raw material for evolution. So in the process, that is, as we have seen in the process of PARVATI, the process crossing over occurs where there is exchange of the segments. During that exchange of the segments, the genetic material is being completely changed. Sometimes your siblings, your brothers and sisters are not similar to you because the crossing over occurs where there is exchange of the segments. So this exchange of segments is nothing but the variation in the genetic material, which is a raw material for the evolution. But the next is that, similar to that of the concept, that recombination and exchange of segments leads to evolution with change in genetic structure. So there is recombination, there is combination of the genes, while during crossing over there is exchange of the segments. That is nothing but an evolution. It will lead to an evolution with change in the genetic material. So these are the significance of meiosis. Then in the next we will go with the concept of differences between the mitosis and meiosis. What are the difference in mitosis and meiosis? Mitosis and meiosis differences. What are the differences of mitosis and meiosis? Mitosis, it occurs in somatic cells, while meiosis occur in germ cells or the reproductive cells. Already you know about the somatic cells and the germ cells. The body cells is called as somatic cells and the reproductive cells are called as the germ cells. Here single nuclear division, and in meiosis two successive nuclear division. We have two phases in meiosis as meiosis 1 and meiosis 2, where the nucleus divides twice, two successive nuclear divisions, while in mitosis there is only division of only once. The nucleus divides only once, so it is single nuclear division. This is known as equational division, while meiosis is known as reductional division. Already we know about it, what is equational and what is reductional. So once again, what is equational? The daughter cells that are formed, the chromosomes are equally distributed to the daughter cells. While in meiosis, there is reduction in the number of chromosomes. They differ from that of the parent cell. Usually parent cells are diploid while the daughter cells are haploid. So we can go with the another point that daughter cells are diploid usually in higher group of plants and animals. Here daughter cells are haploid. The daughter cells are haploid. And prophase is short, while in meiosis prophase I is lengthy with 5 sub-stages. We have learnt there is a stage called as prophase. In meiosis, that process of prophase is very short, while in meiosis, the prophase I is lengthy with the 5 sub-stages. While the other processes where you can see in that process is that the chromosomes during the anaphase, yes, when the separated chromatids, when during anaphase, when they get separated, the separated chromatids are identical, while in meiosis the separated chromatids are non-identical. They are not identical. So when there is no processes like crossing over or synapses or chiasmata, so there is splitting of the chromosomes and the chromatids get separated and which are identical. But as a crossing over occurs, there is exchange of segments. So when the chromatids get separated, they are non-identical. So in meiosis, again we have got these 6 points with the remaining points. Processes like synapses is absent. Crossing over and formation of chiasmata these are all absent in the process of mitosis. So synapses, that is pairing of homologous chromosomes, crossing over, exchange of the genetic material or the segments between the non-sisterchromatids of the homologous chromosomes, while the chiasmata, the point where crossing over, that all stages you will not find in the process of mitosis, while these all processes are present in the meiosis. The main important process for the exchange of the segments, change in the genetic makeup of the DNA. So the processes are present in mitosis while they are absent in the process of meiosis. Then we can say that here, as the mitosis proceeds, so in some cases you will find that there are in the process of prophase, here the chromosomes are not clearly visible during prophase. In prophase, why? They are clearly visible in prophase 1. In the prophase 1. So chromosomes, you cannot see them clearly. In mitosis, you can see clearly the chromosomes at the metaphase stage. But in meiosis, you will see clearly the chromosomes during the process of prophase 1. And here, one daughter cells are formed. Here, two daughter cells are formed. Yes? Sorry. In the process, two daughter cells are formed. Here, four daughter cells. Okay? Only the once nuclear division occurs. When one nuclear divisions, that leads to formation of two daughter cells. Here, nuclear division occurs twice. That it will lead to four daughter cells. So these are the differences, where some differences have been clubbed with each other. Yes. So these are the differences between the mitosis and meiosis. So we'll recollect with the topic. First is the cell division. What is the definition of a cell division? How you will define reproduction, cell reproduction. So what is cell reproduction? Then afterwards we will go with the types of cell divisions. The types of cell divisions, a 3 marks question. What are the types of cell division? That is mitosis, amitosis, mitosis and meiosis. Then followed by the cell cycles. What is cell cycle and what are the events or what are the stages that appear during the cell cycle? So in that stages we have learned what all events happens during G1 phase, S phase and G2 phase. So clearly it can be asked for 5 marks. That is, what is cell cycle and writes its events occur during the various stages. So you have to write the definition of the cell cycle with the events that are occurring. Or for 2 marks they may ask you what are the events that occur during G1 phase. So in G1 phase what all changes occur or what are the events occur. Then in S phase, which is a very important phase in the process. So what are the events that occur during the S phase and the G2 phase. After G2 again what are the events. So this question can be asked for 5 marks or particular events of G1, S or G2 with 2 marks questions. Then it enters into the mitotic division. Yes, in the mitotic division again it starts. In mitosis it is prophase, metaphase, anaphase and telophase. For 5 marks the whole, or for each of them can be asked for 2 or 3 marks. What are the events that occur during prophase? So what happens during prophase? Then what happens? What are the events occurring in metaphase? What are the events occurring in anaphase, and what are the events occurring in the telophase? So just go through that properly. What are the events that occurring in the process of mitosis? So all the events of the mitosis, what happens in the particular events. Then finally there is karyokinesis. 1 mark questions like what is karyokinesis and what is cytokinesis. So as I said that the cytokinesis is division of the cytoplasm, where there is formation of 2 different processes occur in the animal cell and the plant cells. While in the animal cell it is in the formation of a cleavage furrow, while in the plant cells there is a formation of a cell plate. So though it occurs in both of the plant cell and the animal cell, but in plant cell it is by fragments of endoplasmic reticulum and Golgi complex with the vesicles that lead to the formation of the cell plate and divides to form the 2 daughter cells. Then in meiosis we have learned that what is meiosis. In that prophase I is very important, where you have to be explaining or you have to be perfect with the concept of prophase I. We have dealt with the substages of prophase I. The substages of prophase I are leptotene, zygotene, pachytene, diplotene and diakinesis. So what all the important events? If you take the process of leptotene, mainly the condensation of chromosomes occurs, where they appear beaded-like appearance to form the chromomeres. While after leptotene it is zygotene, where 3 processes are important. One is what? There is formation of homologous chromosomes, synapses and synapses homologous and synaptomal complex. So there is formation of that. There is overlapping, crossing over, everything occurs in the process of pachytene. In pachytene, crossing over occurs, there is exchange of the segments. When there is exchange of segments, you can see it brings to variation. That itself is the evolution. It's a process of evolution. While in diakinesis and diplotene and diakinesis, you can see that slowly the chiasmata dissolves. There is a process of terminalization. At the end, there you will see that again that is all the prophase stage. Then it enters to the metaphase, anaphase and telophase, first division with cytokinesis, 2 daughter cells which are haploid. Then and next it enters to the second nuclear division with the same pattern of process but change in here. As I said, due to crossing over, what happens that when there is crossing over, as I had dealt in the some of the points there, the chromatids that get separated are not identical. They are not identical because of the exchange of the genes. So in that we have seen that at the end 4 daughter cells are being formed, while in mitosis 2 daughter cells are being formed. So that is the end of the chapter, cell cycle and cell division. So for the diagrams you can just refer the NCERT textbook, go through the diagrams particularly. They have given you clearly what all the stages are there in that what happens during prophase, what happens during metaphase and all. Here these are all diagram based questions. So diagrams are necessary. You have to be, when you are writing attempting any of the answers, you have to attempt with the diagrams. If the question is asked, explain metaphase, what happens or what are the compulsory, there is a process of diagram. Or what is cell cycle, that is compulsory. So these are all diagram based questions. So go through that diagram based questions. So that ends the topic, the cell cycle and cell division. Thank you.